Hello, welcome to Biology for You. In this video, we are going to discuss this Avery MacLeod and Mac experiment. In this series of videos, we have been discussing different concepts in the molecular biology, and in this very video, we are going to have the basic idea of what exactly is the Avery MacLeod and Mac experiment. Basically, in the previous video, we have discussed Griffith experiments. This Avery MacLeod and Mac experiment is basically an extension to this Griffith's experiment. So, coming to the experiment, this is actually carried by three scientists: Oswald, Avery, Colin MacLeod and MacLeod Mac. So these are the three scientists who developed this experiment, and in the year of 1944. Basically, as we already discussed, this is an extension to Griffith's experiment. So in the year of 1928, which is previous to this experiment, in the year of 1928, scientist called Frederick Griffith, he experimented with the streptococcal pneumonias. Basically, Griffith wanted to learn what exactly is happening when in the case of inheritance. That means he wanted to learn what is the nature of inheritance. So, in his experiments, Griffith used streptococcus pneumonia bacteria, which is present in the R and S forms. These are the two strains. In the case of R strain, which is also called as rough strain, it cannot cause infection in the mice because R strain is lack of polysaccharide capsule on it. But the S strain, which is also known as smooth strain, which is able to cause the infection in the mice, and it is having the polysaccharide capsule outer cover on it. So, due to the presence of polysaccharide capsule, it is able to cause the infection in the mice, and the mice eventually dies when it is treated with this very smooth strain. In Griffith's experiment, he allowed the infection of the R strain and S strain with the mice, and he observed when he treated mice with R strain, it eventually alive. That means R strain unable to cause infection in mice, and it is alive. When he used smooth strain to infect the mice, it eventually died. And in the next step, he used heat killed smooth strain along with live R strain. So, in combination of the heat killed smooth strain and live R strain, when he allowed the mice to be infected with such combination, mice eventually died. And he concluded this experiment with saying that something got. So, there is some change occurred in the alive R strain when it is used along with this heat killed smooth strain. That means some substance from heat killed smooth strain got transferred, is responsible for the transformation of the live R strain into a virulent strain that caused the infection in mice, and mice eventually died. So, he concluded his experiments with saying that there is something called transforming principle. So he said this transforming principle is responsible for the conversion of R strain into virulent strain, and that caused the infection in mice. And he don't know, this Griffith never know what exactly the nature of this transforming principle. And eventually scientific community want to learn what is the nature of this transforming principle. So these three scientists, Oswald Avery, Colin MacLeod, and the MacLeod Mac in the year of 1944 extended this Griffith experiment using these two streptococcal strains R and S. But they did the similar experiment in which the smooth strains are heat killed and R strains are live. So when they used this kind of combination to infect mice, they observed the mice is died. But here they want to know what exactly is the causative agent that is transferred, that is the transforming principle that caused the change in R strain, which is now called as a virulent strain and infected the mice. So they want to learn what is the nature. Basically, there are three kinds of macromolecules, that are DNA, RNA, and proteins. So either one of them is responsible for the transformation of the rough strain into smooth strain, or rough strain into a virulent strain that causes the infection. So they want to know what exactly among these three, what exactly is responsible for this thing. So they want to know that, and they did experimenting with this. So in first experiment they used proteinases, which are the enzymes which destroy the protein. So they used the heat killed smooth strain, they treated with the proteinases, and they showed that the mice is still died. That means even the smooth strain is killed, the smooth strain is treated with the protein degrading enzymes like chymotrypsin or trypsin. The mice is able to be infected with this R strain, and that is died. That means the transforming principle which is responsible for the change of the R strain is not the protein. Then in the next step they used RNAs, or ribonucleic acid degrading enzymes, or RNA degrading enzymes. So they use this kind of enzymes to degrade the RNA. So in heat killed smooth strain, the RNAs are degraded, and when they used these kind of RNA degraded smooth strain with the combination of rough strain, the mice it still died. That means this RNA is not responsible for the conversion of rough strain into virulent strain. And in the next step they used deoxyribonucleases or DNases. When they used DNases to treat with the heat killed smooth strain, so in this heat killed smooth strain, all DNAs are degraded, and they used in combination of rough strain, the mice is alive. In this case they observed that the mice is alive. That means the R strain in this case is not able to cause infection, it is not transformed, or this R strain is not converted into virulent strain because of the absence of DNA. So this DNA is not transformed from smooth strain to the rough strain, and rough strain it remains the same non-virulent strain, and it is not able to cause infection in mice, and the mice is alive. So this is the basic experiment that done by this scientist, and they concluded that DNA as genetic material. but the scientific community is not actually accepted such experiment, and basically they also try to explain it by studying the nature of the transforming principle. So somehow they managed to isolate this transforming principle, and they studied the nature of the transforming principle, that is number of nitrogens, number of phosphorus and all chemical structures present in it. So these number of atoms present in this transforming principle is similar with the number of nitrogens or chemical compounds present in the DNA. So this is the other support provided by the scientists, that the nature of the transforming principle is similar with the DNA, and the number of chemical structures present in the chemical atoms present in the transforming principle are similar with the DNA. So they concluded that DNA as a genetic material, but generally this experimentally, the DNA as a genetic material, the concept is experimentally proved by the next experiment in the 1950 decade, that is Hershey and Chase experiment.